welcome everyone. It's great to see you all. Thank you for joining us for this second edition of Sun and Science. Those of you who joined us last week will have heard that this is a new version of something that I can fairly say is famous within the Faculty of Science. In my role as Dean of Science, I've been able to host Soup and Science now for the last 5 years. What we're doing is we're showcasing the research that happens in the Faculty of Science, and in particular showcasing the fact that one of the most important parts of the undergraduate experience in the Faculty of Science is the opportunity to engage in real science. And by that, I mean science that is far beyond washing test tubes or doing data entry. Every undergraduate student in the course of their degree will have the opportunity to have some real meaningful experience in research, working beside PhD students, postdoctoral fellows, and professors, such as the professors who will be speaking today. The format is quite simple. We'll have 3 speakers. Each will be speaking for at most 5 minutes. Very challenging for professors to speak only for 5 minutes about their research, but nonetheless they will, or they'll be cut off. And then we'll have an opportunity. You'll have the opportunity to meet them in breakout rooms, and that process will be described in a few minutes. But let's start with the presentations, because that's why we're here. And we have 3 speakers, as I said, Professor Adrian Liu from Department of Physics, Professor Christie Rowe from Department of Earth and Planetary Sciences, and Professor Janine Mauzeroll from the Department of Chemistry. So we'll start with Adrian Liu, and it's all yours, Adrian.
Great. Okay, well, thank you very much. Oops, I gotta do this again. There we go. So I am very excited today to tell you about some of our efforts to make big maps of our universe. So one way to talk about my research is just we are making a big map in space and time to literally observe parts of our cosmic history. And in particular, something that I'm really interested in is to, of course, this happens now, is to understand how the first generation of stars and galaxies form. So for instance, here is a movie showcasing what happens when the first galaxies start forming. And what you're seeing in these blue bubbles are ionized regions. So as these first galaxies form, they give out UV radiation that then ionizes the hydrogen in the universe, and eventually more and more of the universe gets ionized, and there's a dramatic transformation that takes place. Now, the really interesting thing about this is that nobody's yet seen this. We would like to be the first ones to actually build a big radio telescope in South Africa to do this. Construction is not yet complete, but when it is, this will consist of 350 14-meter dishes that act in concert as a giant super telescope to help us detect faint signals from hydrogen around the time the first stars are forming. Now, you get data from a cutting-edge instrument like this, and it tends to look something like this, where you end up with, oops, that didn't work out so well. Second, yeah, here you will see, for example, that what I'm doing here is I'm showing you data from each of these individual telescopes. And if everything were working properly, because I've got many identical copies of these mini telescopes, everything should look the same. So the question is, how do you make bad data like this look something more like this, where everything that should be the same does look the same? And the real answer to this, it sounds a little silly, but it's true, is just to have really great students working on this. And so, for example, here are Deacine, Ronan, and Taylor, all of whom are physics undergraduates who've helped massage our data to make it all turn out right, to calibrate it, and so on. But even after that, there's a lot of work to do, right? The data looks good, but what can happen is that if you are interested in looking at tiny, tiny cosmological signals, it might be contaminated by lots of really bright radio emissions from TV stations, from radio stations, from our own Milky Way galaxy, in fact. So here's an example of that. And so one of the things we have to do is to filter out our data. But one of the annoying things about filtering data is that it can destroy some of the information you're looking at. So on the left here, I've got an example of one of those bubble maps you saw in the movie earlier. On the right, you've got an example of what happens after you've filtered this data, chucking out the contaminants. And what you see is that I've lost a lot of the resolution that I would want for these ionized bubbles. But we're finding that maybe machine learning can save the day, reconstructing some of these shapes. And this is yet another example where this was really undergraduate-driven research. Damien, Christine, and Sam were the real drivers of this kind of stuff. And going further down the pipeline, if you think that you've cleaned out your data properly, you've recovered a lot of the lost information that you might have accidentally filtered out, how do you connect this to theory models? And again, this is just a matter of a lot of hard work. You know, science is a lot less about the magical inspirational moments as lots of hard work, a lot of which is, again, done by undergraduates like Joel and Henry. And so, for instance, Henry did a very accurate, precise calculation of our uncertainties in some of the theories that were fitting to our data. And he's finding, for example, that a lot of published papers are too optimistic by a factor of 2. So in conclusion, I think the message I'm trying to get across is that they're really exciting times. These are really exciting times. We're using these big radio telescopes to measure light from when the first stars are forming. And a lot of this work really is driven by undergrads. It's kind of strange that we sometimes call undergrads research assistants, because really they're the ones doing the real work here. Thank you.
Okay, so next we should have Christie Rowe. Sorry, Christie, let me just... Christie, are you able to click on the screen?
Yeah, does that work?
Yeah, okay, perfect.
Yep, okay. Okay, hi everybody, oops. Oops. Is that? Okay, my name is Christie Rowe. I'm a field geologist. That means I study rocks. And my particular specialty is understanding how rocks cause earthquakes, how earthquakes happen in geological faults, and what we can learn about the rock record of earthquakes tells us how the earthquake cycle works in the earth, and what kind of past earthquakes have happened in an area, which then can go into hazard estimations for understanding future earthquakes. So I thought I would show you today an example of a past earthquake. So this is Japan, and the little blue arrows are GPS stations moving horizontally, and the red arrows are GPS stations moving vertically. So that big disturbance you just saw was the 2011 magnitude 9 Tohoku Oki earthquake, and there'll be an aftershock, magnitude 7 aftershock here in a minute. Wait for it, there it is. Okay, these GPS stations collect the position of the Earth's surface relative to satellites once every second, 1 hertz. So the long period waves from the earthquakes are actually picked up on the GPS stations. Oops. So I'm going to show that once again now that you know what you're looking at. If I can. Oops. I'm clicking too eagerly. Let's see. Sorry. There's a little bit of a delay in here. Is it running? Yeah, okay. So this I really like this movie because it tells us, it really expresses what humans feel when an earthquake occurs. It shows not only like the big jarring effect of the surface waves when they come from the epicenter of the earthquake, but it also shows the permanent displacement of the land surface. So in that magnitude 9 earthquake, Japan moved several meters east relative to the global reference frame, and the coastline of Japan dropped by almost a meter relative to the global reference frame. So these are permanent deformations of the Earth's surface that affect things like local sea level and infrastructure, and they can cause a lot of damage. So what my research tries to do is to go get kind of granular about this. Sorry, I keep clicking too eagerly. I'm going ahead. So this is from a much smaller earthquake. There's a magnitude 6 earthquake that happened in Napa County, California, that was on a fault related to the San Andreas Fault. And we have a nice markers here, which we don't always have in geology. So we have a white paint stripe going down the middle of a street, which presumably was straight before the earthquake. The day of the earthquake, which is the picture on your left, it was displaced about 10 centimeters. So that is the actual amount that the Earth's surface changed shape in the earthquake. Then a day after that, it had very slowly crept like another 10 centimeters. So on the earthquake itself only lasted for a few seconds, so it was a jarring 10 centimeters, and then there was a creepy, quiet, no seismic waves produced 10 centimeters that happened over the next day. So now we have twice the displacement. So this is a clue that like the earthquakes aren't the only thing that's happening when these faults are chugging along. There's actually a lot of things that are happening in between earthquakes that are silent, that the rocks are doing that we don't necessarily think about or know about in our everyday life. So I'm going to show you just like a quick example of student projects that have happened in my lab. So this is a study of a fault in South Africa that's very like the San Andreas Fault, only it was like the San Andreas Fault about 900 million years ago. It's no longer active, but in that time, 10 or 15 kilometers of rock has been eroded away. So the part of the fault that you can see if you go out on the rocks today is the same as the part of the fault that's currently controlling the San Andreas inside the crust today. And this is a diagram that came out of the work of about 3 or 4 students, PhD, master's, and undergrad students, trying to figure out all the parts of this machine and how they work together under the ground. So the way that we do this is a Google Earth image, and you can see the 2 kilometer scale bar at the lower left. So this is 10 kilometers area. You can see in the beautiful arid climate of South Africa that the rocks are nicely exposed at the surface, and we don't have any of this organic matter to get between us and the good stuff. So you can see the scar cutting across the landscape. That's the actual trace of the fault. And whereas in the Napa earthquake, we saw only a tiny crack a few millimeters wide, that's what we see at the surface. But the actual zone where the earthquakes are produced is a few kilometers wide at depth. And this is what it looks like when you get down inside it. So my students went out there and they went and walked out many parts of this fault system and made structural and chemical maps of all the components of rocks that are involved in these processes. And through this mapping, should go to a small photo now. Yeah, you can see the tiny layers and the little black smears in that rock photo that's on the left side of your screen are actually fossilized earthquakes. These are little black bands of melted rock because the stresses are so great when the rocks are rubbing by each other in an earthquake that it causes frictional heating that can actually raise the temperature by like 800 degrees for a few seconds and melt a tiny sliver of rock. So there they are. And when we map these using some of the equipment that we have here at McGill, so this is a map of the sodium content inside a tiny area of that rock, and you can see 500 micron scale bar at the lower left. The albite grain is a feldspar that's high in sodium. There's a big yellow circle in the lower left corner, and that grain partially melted and got swept into this melt layer during the earthquake. So that shows the way the rocks were flowing. And the melt was very slippery during the earthquake, so it facilitated the earthquake. Okay, here we are in Quebec, and this is a map of historical earthquakes. It was made by a student here in the department, Andreas Peña Castro. So these are earthquakes in about the last 150 years or so in Greater Quebec, around Montreal. And you can see there's a lot of magnitude 6s and a few magnitude 7 earthquakes that have affected this area in the last 150 years. So over 150 years, that's nothing to a geologist. that's a really short period of time. To humans, that feels like a long time. And Montreal was built during this time period. So 150 years ago, there wasn't really too much infrastructure here. And now we have this huge city. So we're very concerned about making sure that we have skill sets across Canada in lots of different fields in engineering, sciences, urban planning, and business and government, where people understand a little bit about earthquakes, how they work, and how Canadians need to be prepared for this. So we're currently engaging in a big training program proposal to try to get lots more people exposure to earthquake science. Here's an example of a geologic map of Montreal, and each of these colors represents another different rock type, and the little black lines where you can see the divisions between the colors where they're offset, those are faults. Nobody knows which ones of those faults have been recently active, if any. So there's a lot of work that we need to do right under the city right now. And if you're interested in getting started on learning the skills to do that, I'm teaching a class in the fall called Geology in the Field. It's there's no experience necessary. It's for people that don't have any geology background or have just a little and just want to learn about how to take notes in the field and how to be observational field scientists. And because I love earthquake geology so much, we'll probably talk a lot about that. But anyway, I hope I get to meet you guys all soon, and I'm not sure about my time, but I'm done. I can't hear you, Preeti.
Sorry. Okay, thank you so much. So next we have Professor Janine Mauzeroll from the Department of Chemistry.
Are you gonna let me share my screen?
Yes, yes, yes. Janine, you should be good to share your screen.
Well done. Well, it's my first Soup and Science remote. And as expected,
There we go.
You know, it turns out to be exactly at the same time as my kids' clarinet and French horn class. So if you guys hear clarinet or French horn, please know that it's all part of the remote fun of science and life at home. So my name is Janine, and I'm a prof in the chemistry building, and I'm an electrochemist, which means I love everything that has to do with electron transfer reactions. So if I see if this works, can you see my second slide? Yes, I see. Okay, wonderful. So I'm particularly interested in microscopic electrochemistry. So can you guys see my pointer? Yes, okay. So here I'm pointing to a very small microelectrode that we make in our lab. It's roughly 1/4 the diameter of your hair. And it allows you, when you connect it with high positioning motors, it allows you to position it nearby surfaces, like surfaces that are undergoing corrosion, solar cells, any kind of catalytic material or biological material. And what you can do with this technique is quantify how fast the electrochemical reactions are occurring at your surface. So in our group, we apply this technique to materials that degrade through corrosion. We work on materials with batteries, for example.
Can everyone hear me? Okay, so let's just give Janine a second. It looks like she her computer may have frozen. Oh.
You guys lost me?
Yes.
You guys lost me. All right, well, let's try again. See, it's nothing if not fun. Can you guys see it again? Here we go. Okay. And so I'm sharing this story. I don't know if I already told you this or not, but I'm sharing this story because it's a great success story with regards to an undergraduate. Okay, so the whole premise behind the work is that we're trying to use these small microelectrode to track the cell metabolism or communication, right? Cells end up having a lot of transmembrane proteins, and they secrete a lot of molecules. And thankfully for us, a lot of those are electroactive. And so we apply these methods either to study cancer cells, to see how resistive or how strong the phenotype of the cancer is. Oh my God. Or we use them to study. Did you lose me again? Pretty? No, okay. We use them to study neurons. I'm sorry, the screen is like flicking on and off. And that's the story I want to talk to you about today, where we design these biosensors to detect a molecule that's called D-serine in the brain of animals. And so neurons are notorious for secreting a lot of molecules that are electroactive, like dopamine. But there are lots of neuromodulators and neurochemicals that in fact are not electroactive. And D-serine is one of these molecules. You see, it doesn't really have anything to undergo electrochemistry. And so what happens is that we need to use the power of electrochemistry combined with enzymes. Now, D-serine is a really interesting molecule because it affects synaptic plasticity, and it also is involved in a lot of pathological processes such as schizophrenia, aging, or neurodegeneration. And so here's what Annie, who was a neuroscience undergraduate student, did. She came to see me. She was in like U0, and she's like, I want to do electrochemistry, and I have zero experience. And I said, well, that's fine. And so what she did is she developed this microelectrode. Here you see in the dark gray, it's the glass. And in the dark black, it's the platinum. That's the reactive part of our sensor. Now, in the brain, there are a lot of molecules that are electroactive and that could generate a current that would skew our results. So what we need to do is electrodeposit this blue layer, which is a polymer-based layer. It basically blocks off large interferences.
Okay, so let's just give that a second. Oh. Janine, you want to try again?
At what part did you lose me?
At the, I think it was the third slide. Yeah, this one.
This one?
Yes.
Okay, starting again. So now we got the platinum, that's our reactive electrode. We got the blue part, that's our polymer layer, that's preventing all the molecules from making it and skewing our results. And then we have the DAAO, which is an enzyme that's specific to turn over D-serine, okay? So what happens is when a neuron is secreting D-serine, it's gonna get turned over by the enzyme and it's gonna generate hydrogen peroxide. Now, hydrogen peroxide is a wonderful molecule. It's very small. It can get through my polymer layer and to my detection zone. And that's where the current's going to be coming from. And so now, meet my experimental sample. Here you have Xenopus levis tadpole, which are the organisms with which we work. Now, we use them because, as you can see, they're transparent. And it's very easy to locate the neurons that we're particularly interested in. It's also quite quick to get from an egg to a tadpole. It takes about 1 week. And so here's the area of the brain that we're interested in. It's called the optic tectum. its diameter is roughly like 200 microns. So then what we do is we locate the neurons, we position our biosensor, and we're going to want to track in vivo the release of D-serine. So what you see here is you have a graph where you have the current. That's my hydrogen peroxide current that I told you was coming from the fact that the D-serine was being regenerated by the enzyme. And on the y-axis, you have time. So if we move along, here my biosensor is positioned next to the neuron, and it's basically in buffer. Then we add injections of solutions of different concentrations of D-serine. That's important because we need to calibrate our biosensor because we're in the business of quantifying concentrations. And so you see here that I have a nice linear relationship between the current that I'm measuring at my biosensor and the concentration. And then I end up being able to detect the in vivo concentration of D-serine in the optic dectum of tadpoles. Our goal now is to now measure in real time the D-serine release from different types of neurons, because there's a discrepancy in the field. People don't understand which neurons store D-serine, which neurons are responsible for its release. And since it's tied to schizophrenia and depression, it's really important that we understand the source and the extent to which different types of neurons can release D-serine. So as I told you, you can't do this without a great team. And my team has always involved undergraduates. And the story that I talked to you about today involves Annie, which was a U0 neuroscience undergrad. So I highly encourage you to seek out profs and research experience because it's really fun and you might get to learn to play hockey.